So looking at reproduction, Selaginella is similar with Lycopodium, where it reproduces vegetatively and by sexual methods. So in the vegetative reproduction, they take place by tubers, okay? They're tubers, buds, and through fragmentation, okay? Bulbils and dormant buds are produced in the aerial branches, while tubers may be aerial or underground. In the favorable condition, they will germinate to produce new sporophytic plants. In Selaginella rupestris, the trailing branch of the stem develop adventitious branches that separate from the parent plant and grow into new plant body. Okay, so simply just by the fragmentation, the buds, the bulbils, tubers, all these will just reproduce and develop into a complete new plant. Okay, the bulbils and the buds, they can come above the ground, aerial, and the tubers, they can come above ground or below ground. Okay, then in rupestris, there are branches which develop in the side, adventitious branches, and they will just break off and develop into a new sporophyte. Then let's look at how it reproduces by spores. Okay, so haploid spores are produced in the sporangium. There, this one. The sporangium are located on the sporophyll, and the sporophyll are compactly arranged in the form of cone or strobilus. So this one you already know from the theory. So I'll just read out. All the species of Selaginella, they will form the cones. Generally, the strobilus occur terminally on the side branches, but in some species, the apical meristem of the cone may continue meristematic activity, producing foliage leaves, and therefore produce a shoot with sporophyll. Okay, I have to explain only. Okay. So all the species of Selaginella, they will produce the cones. Okay. Okay. The cones usually they occur at the tip of the leaves or on the branches at the tip, on the side of the branches here at the tip. Okay, but in the side. All right. But in some, though the tip part, the apical part, it will continue to form normal cells, and therefore it will produce a shoot with the sporophyll. Okay, means instead of forming a cone, no, like this, it will continue behaving like a leaf. It will form sporophylls, leaves, but on the sporophylls will be the sporangium. Okay, so there are two types of spores. So it is heterosporous, and the sporangia are of two types, that is microsporangia and megasporangia. The sporophylls associated with these two types are known as microsporophyll and megasporophyll. So I've already explained that during practical. So it's very easy for you, no? See the image. There you have the ligule, you have the sporophyll, then this one the megasporophyll, this one is the megasporangium, these are the megaspores. Then if we look up, 1 minute, you can see the microspores with the ligule, the microsporangium. Okay, so we have already drawn this. This is very easy for you. I've already explained this. I've already explained that. Okay, so let's look directly at the sporangium. The mature sporangia, they have a stalk and a two-layered jacket. Okay, the microsporangia are slightly elongated and reddish to bright orange in color, and the megasporangia are larger and are lobed. Okay, like a lobe like this. Okay, the megasporangia are whitish yellow or orange in color. So you have to remember yellow or orange, the big one, and the small one is red to bright orange. Okay, so the deep colored is the small one, micro, and the yellow one, the lighter color, is the big one, the megasporangia. So these are the images. This one you have already drawn in your practical. So you have a two-layered microsporangial wall. You can see the microspores. You can see the ligule. Here you can see the megaspores. You can see the wall. You can see the ligule again. Okay. So let's look at the development of the sporangia. Okay, so first the micro and megasporangia in the beginning, they will develop in the same way. Okay. That is, their development is of the eusporangiate type. That is, the sporangium developed from a group of initial cells. Like we did in Lycopodium, remember? We always start with an initiating cell. Okay. So see here in the image, as I said, first, if you draw first, no girls, very easy for you to explain from the diagram only. So while you are studying, please hold a pen or a pencil, whatever, and paper, and you draw. Okay, as you're studying, you draw, you practice the diagram. So we see the sporangial initials here, there, and they come and they divide, and then we have one layer of archisporal initials and jacket initial. So obviously the jacket initial will form the jacket layer, and this archisporal initial will form the sporogenous tissues. So here we can see nicely the jacket layer and the archisporal cells. Then this archisporal cells will become into the sporogenous tissue, and then from this sporogenous tissue we can see one layer of tapetum. Tapetum is a nutrition, okay, it will give nutrition to the sporogenous tissue, and then above the tapetum is the jacket cells. Okay, so it's like this: jacket, double jacket, then tapetum, this one, and then sporogenous tissue inside. Okay, so it'll be very easy now. We look at the text. The site of the sporangial initiation is in the superficial cells of the axis directly above the sporophyll. Okay, means on the surface of the leaf. Okay, then they will divide periclinally into an outer jacket initial and an interarchisporal initial. From here, like this one and this one, JI and AI. Okay. Then we get a two-layered sporangial jacket formed by repeated divisions. So it will keep on dividing. Then again, those archisporal cells will produce outer and inner layer of cells. Then the outer layer of cell will become into the tapetum, and the inner cell, they will become into the sporogenous tissue or the spore mother cells. Now the sporangium at this stage will consist of the immature sporangial wall of two layers, a short stalk and a tapetal layer which contains the sporogenous tissue. Then from this stage, they will develop into different ways accordingly, whether it will develop into microspore or megaspore. So now let's see how it will develop into the microspore and the megaspore. So further development of the sporogenous tissue gives rise to the microspore and the megaspore. In the microsporangium, about 80 to 90% of the sporogenous tissue will undergo meiosis and form tetrads of haploid microspores. Okay, so this one they will decide by themselves. At first, in the initial, they will divide the same way. You will not know which one belong to which. But slowly, after they have formed the spore mother cells, they will decide by themselves who will become the megaspore, who will become the microspore. So in the microspore, simply all those cells, they will undergo meiosis. They will form tetrads, which are all haploid. Okay. Then remember I told you they were triangular in shape, the small ones. Okay. More or less tetrahedral in shape. Okay. Triangular means tetrahedral, no? Just like that carbon tetrad, no? Like this. In physics or chemistry, whatever you did. Small in size, and they have outer exine and inner intine, obviously. Okay, then let's go back to when we stop in the sporogenous tissue. Now when it will become the megasporangium, only one will become functional. Okay, so in a potential megasporangium, only one spore mother cell becomes functional. Okay, The remaining non-functional megaspore mother cells will develop large vacuole and accumulate starch, while the functional megaspore mother cell will retain a dense cytoplasm which is rich in RNA and lacks starch. Okay, so this one is going to become the egg, no? So it will be very dense cytoplasm, means they will have lots of organelles, lots of nucleus, right? Will be very, very rich. Okay, but those non-functional one, okay, if this is the spore mother cell, so for up to here, the spore mother cell is the same, no? This one, if it wants to become the microspore, it will develop meiosis like this. But if this spore mother cell want to become a megaspore, these will become non-functional, and this one, last one, the functional megaspore, it will become very dense cytoplasm, lots of RNA, lots of DNA, lots of cytoplasm, lots of organelles, while this one, the non-functional one, will have lots of vacuole, okay, means empty space. Okay, then the functional megaspore mother cell will undergo meiosis, forming 4 megaspores, and then those non-functional one, they will degenerate. So one spore mother cell, it will undergo meiosis into 4. Out of that, these 3 will die off, they degenerate, while this functional one will remain. Sometimes more than one megaspore mother cell becomes functional, resulting into 8 or even more megaspores in a single megasporangium. Okay, so it's not exactly one, sometimes it can be more. Then this megaspore will develop a thick cell wall, and it will have lots of spines and ridges known as exine, and it will have a thin intine. Okay.